So if you had inky water, for example, and the ink in the water would be here, the water would evaporate up and condense here, coming out as distillate, so just like filtrate from the previous one, and then the ink would be left behind. So that's distillation with the full apparatus. So the last one, explain how chromatography can be used to separate substances. So the classic example here is if you've got different coloured inks and you want to separate them out. So there are various parts of the chromatogram that you need to know about. Here we've got the normally this is water in the chromatogram, but this formal name of this is the mobile phase. Mobile because it moves, and then here this is a piece of paper, normally filter paper. In this case it's called the stationary phase. The line in which you put your samples on, that's called the baseline, and you put your samples on the baseline. And if you are analysing ink in water, you would normally draw this in pencil. Otherwise it's going to run when you run the chromatogram, and then you allow the mobile phase to soak up the stationary phase, in this case the filter paper, and depending on the solubility of the different inks, they will dissolve well within the mobile phase and they will move faster up the paper than the other ones. So if we were looking at the pure samples here, the blue sample, the green sample, the red and the yellow sample, red is the most soluble in water, so that's most water soluble, as it moves the furthest, and the one that moved the least is the green one, so that's the least water soluble, and then the yellow and the blue are in between. So pure samples, pure substances will give just one spot on the chromatogram. If you've got a mixture like number 3 here, and it's a mixture of the green and the blue and the yellow and the red, then you will get as many spots as there were substances in the original mixture. And we can tell whether they're the same substance if they're coloured. Obviously we can compare the colour, but if these weren't coloured, if these were other chemicals without colour, we can develop the chromatogram in different ways with other chemical substances or UV light, and if they are at the same height on the chromatogram, then they are likely going to be the same substance. So these two here are the same as the same distance moved. Okay, so analysing chromatogram to identify different substances, you look for spots at the same height. So that's a quick run through of some of the basics of separation techniques. Obviously this is just a quick overview. You'll need to do some more work to have a good understanding of it. BBC Bite Size has good notes. You should have good notes from your lessons, and then there's lots of places you can try out some questions, such as the BBC Bite Size, or log on to Caboodle or Sam Learning, have a go at some of the questions there. So they're the websites for those ones. And the reason Mike Curtis, this one kept popping up, is this was one of the websites I found a picture on, and he's got a little quiz that you can download, which covers lots of Key Stage 3 chemistry, physics and biology. So I'll leave it there for you to go away and do your own revision. Any questions, ask your teacher in class or at any time before the exam. Best of luck.